Imagine you are holding a piece of amber, small, warm, transparent, and inside it, an insect. It died 100 million years ago. At the moment it died, dinosaurs were still walking the earth. The continents stood in different positions. Human beings did not exist even as an idea. And yet, there it is, three-dimensional, with legs, with the structure of an eye, sometimes with traces of soft tissue, not a cast, not an impression. The creature itself, caught in 1 second and stopped forever. Before we get down to business, please subscribe to this channel. Your support makes it possible for us to continue this work, and we are genuinely grateful for it. Thank you. Now, let us talk about time machines. There is a hierarchy of ways the past survives into the present. At the bottom of that hierarchy are things like written records, useful but young, a few thousand years at most, and even then fragile, interpretive, filtered through human memory and human language. Above that, stone tools, pottery, the physical debris of ancient civilizations. Older, more durable, but still mute in important ways. They tell you that something existed. They do not show you what it looked like alive. Above that, traditional fossils, bones, shells, teeth, the hard mineralized parts of organisms that were slowly replaced over millions of years by stone. A dinosaur skeleton is extraordinary. It tells you the size, the proportions, the attachment points of muscles. But the animal inside the skeleton is gone. What you have is an architectural plan, not a building. The flesh, the color, the texture, the behavior, all of that is inference, educated guesswork, the careful imagination of paleontologists working from indirect evidence. And then, at the very top of that hierarchy, in a category entirely its own, amber. Amber is not a fossil in the traditional sense. It is not mineralized bone. It is not a replacement of one material by another. Amber is the original material, tree resin, transformed by time and pressure and chemistry into something permanent, something that neither decays nor dissolves, something that preserves what it captures in 3 dimensions, in exquisite detail, sometimes with soft tissue intact, sometimes with color preserved, sometimes with the original chemistry of the organism still present and readable. Amber is the only substance on Earth that stops time. Not metaphorically, chemically, physically, in ways that are still being worked out. Amber arrests the process of decay at a moment in the past and holds it there. When you look through a piece of amber at an inclusion, at the organism trapped inside, you are not looking at a record of the past. You are looking at the past itself. That is a distinction worth sitting with for a moment, because it changes everything about how we think about what amber is and what it can tell us. We are going to spend the next hour and a half inside amber. We are going to travel from the chemistry of a living tree producing resin to the moment that resin traps something, and from that moment across geological time to the laboratory where a scientist with a microscope is looking at a creature that saw living non-avian dinosaurs. We are going to visit the amber markets of Myanmar and the beaches of the Baltic Sea. We are going to discuss what Jurassic Park got right, what it got spectacularly wrong, and what the truth is stranger than the fiction. And we are going to end up, I think, with a different sense of what it means to hold time in your hand. Let me begin where amber begins, with a tree and with pain. A tree does not produce resin because it wants to. Resin is expensive, metabolically speaking. It takes significant energy to synthesize, and a tree has no energy to waste. Resin is produced for one reason, because something has gone wrong. Something has damaged the tree. An insect boring into the bark, a fungal infection penetrating the wood, a wound from a broken branch or a lightning strike. And the tree is responding. Resin is a tree's emergency medical system and its security force simultaneously. When damage occurs, resin ducts in the bark and wood rupture and release their contents into and over the wound site. The resin floods the damaged area. Its chemical composition is complex and deliberately hostile. Terpenes, terpenoids, phenolic compounds, organic acids, a cocktail designed to do several things at once. It fills the wound physically, sealing it against air and water and further microbial entry. It is antimicrobial, killing or inhibiting the fungi and bacteria that would otherwise colonize a wound site. And it is toxic to the insects that created the wound in the first place, trapping them in a viscous, chemically aggressive substance while simultaneously poisoning them. This is the moment that matters for our story, because resin is also an extraordinarily efficient trap. The physical properties of fresh resin are remarkable. It is viscous, thick enough to hold an insect immediately upon contact before the creature can escape. It is adhesive, sticking to whatever it touches with considerable force. And it flows slowly, so that something caught in one drop may be covered by subsequent flows that preserve it in 3 dimensions. An insect landing on a resin-coated surface, or a small creature brushing against a resin-producing wound on a tree trunk can be immobilized almost instantly. If the resin then continues to flow, the organism is encased. If conditions are right, it will stay that way. But here is the thing about resin. Most of it does not become amber. Most of it just sits on the tree surface and evaporates, or gets washed away by rain, or falls to the forest floor and decomposes. The volatile components of fresh resin, the compounds responsible for that distinctive smell when you cut a pine tree, escape into the air over weeks and months, and what remains is an increasingly solid, increasingly stable substance. But even that semi-solidified resin on the forest floor, in the presence of oxygen and moisture and decomposing bacteria, will eventually break down. It will vanish, taking everything inside it with it. For resin to become amber, it needs to be buried. It needs to be removed from the surface environment where decomposition can reach it, covered by sediment, buried under layers of soil and organic material, eventually compacted under the weight of more sediment above. And it needs time. The process of transformation from semi-solidified resin to true amber, the process chemists call polymerization, takes millions of years. The volatile compounds continue to escape. The molecular structure cross-links, forming longer and longer polymer chains. The material becomes harder, more stable, more chemically inert. The inclusions inside, the insects, the plant material, the air bubbles, the water droplets, whatever was caught in that original moment of disaster, are sealed inside a matrix that is increasingly resistant to everything the geological environment can throw at it. What comes out at the other end of this process, after millions of years of burial and pressure and chemical transformation, is one of the most stable organic materials known to science. Amber can survive for hundreds of millions of years without significant degradation. It is chemically inert, mechanically durable, and, crucially, transparent. You can see through it. Whatever it captured, it shows you. The oldest amber ever found dates to approximately 320 million years ago, the Carboniferous period, the same era of giant dragonflies and coal forests we discussed in our previous video. But the inclusions in Carboniferous amber are sparse and not well preserved. The amber that transformed paleontology, the amber that contains the extraordinary specimens you have seen in photographs and documentaries, is younger, and it comes from two main sources that could not be more different from each other. The first is beneath the Baltic Sea and along the beaches of northern Europe. The second is in a war zone in northern Myanmar. Both are extraordinary. Both are controversial in different ways, and together they have rewritten the history of life on Earth in ways that continue to accelerate every single year. Let us start in the north. Imagine a forest that no longer exists. Not in the sense that it was cut down or burned, it simply has no living relatives. The trees that produced what we call Baltic amber belonged to a genus that is entirely extinct, tentatively classified by botanists as related to modern members of the family Sciadopithiaceae, the umbrella pines of Japan, but not directly ancestral to any living species. This forest existed approximately 44 to 49 million years ago during the Eocene epoch in what is now Scandinavia and the area around the Baltic Sea, where the climate at that time was subtropical, warm, humid, dense with vegetation in a way that Northern Europe has not been since. We know this forest through its amber. We know it the way a crime scene investigator knows a room, not by being there, but by reading everything that was left behind. And what was left behind is staggering in its variety. Baltic amber is estimated to contain specimens from over 40,000 species of insects and arachnids, more than 40,000 species preserved in a material that formed before human civilization existed by a factor of more than 10,000. Beetles, flies, wasps, ants, spiders, mites, book lice, lacewings, moths. The full spectrum of an Eocene forest arthropod community, frozen at a moment roughly 46 million years ago and extracted from the ground today. The Baltic amber deposits are, by geological standards, not deeply buried. The original forest that produced this resin grew along what was then the southern coast of a landmass geologists call Fennoscandia, a combination of what is now Scandinavia, Finland, and the Kola Peninsula. As sea levels changed and the Baltic basin formed and subsided, the amber-bearing sediments were buried beneath marine deposits. They have since been eroded by Baltic sea currents, and for thousands of years, pieces of amber have been washing ashore along the beaches of Poland, Lithuania, Latvia, Denmark, Germany, and Russia. This is not a metaphor or a poetic device. You can go to the beach near Kaliningrad, Russia, formerly Königsberg, after a storm, and you can pick up pieces of amber from the sand that contain 46-million-year-old organisms. This has been happening for as long as humans have lived near that coastline, and they have always known it was special. The ancient Greeks called amber electron, the root of our word electricity, because amber produces a static charge when rubbed, a phenomenon the Greeks noticed thousands of years ago. They thought amber was the tears of the Heliades, the daughters of the sun god Helios, hardened into gemstone as they wept for their brother Phaethon. The Romans knew it as succinum, the juice, recognizing even without chemistry that this was something that had once been liquid. But the Greeks and Romans also noticed something else. They noticed the inclusions. Pliny the Elder, writing in the 1st century of the Common Era, described insects preserved in amber with what reads as genuine wonder, even across 2,000 years. He observed that the creatures must have been caught while alive, trapped in the moment. He was right about that. He had no way of knowing how right. Let me give you some specific examples of what Baltic amber contains, because the abstract statement that it holds over 40,000 species does not convey what it actually feels like to look at this material. In one well-known specimen held at the Natural History Museum in London, a spider is preserved in the act of spinning silk, the silk thread visible emerging from the spinnerets, stretching outward into what was once air and is now amber. The thread itself is preserved. Under a scanning electron microscope, the individual silk strands are visible, each one thinner than a wavelength of visible light. The spider died in the middle of an action it had performed thousands of times. The action is preserved literally in the moment of its performance. In another specimen, a mite, one of the smallest of arachnids, barely visible to the naked eye, is preserved attached to the wing of a fly. The mite was a phoretic passenger, an organism using the fly as transport, clinging to the wing not to feed on it but to hitch a ride to a new location. This behavior, phorecy as biologists call it, is practiced by many modern mite species. Its presence in an Eocene amber specimen tells us that this specific ecological relationship, this precise behavioral strategy, existed 46 million years ago, essentially unchanged. The relationship between the mite and the fly is frozen in amber. The ecology is readable. In a third specimen, from a collection at the University of Göttingen, a fungal infection is visible in progress on a piece of bark. The fungal hyphae are preserved in their act of penetrating the bark tissue. The bark's defensive response, a resin outpouring, is literally the amber that surrounds both the bark and the fungus. The tree was fighting back against this infection, and in fighting back, it preserved the very thing it was fighting. There is an irony in that which I find genuinely moving. These are not exceptional specimens. They are representative of the kind of thing Baltic amber routinely produces. The natural history of the Eocene world, its ecological relationships, its behavioral repertoires, its biological interactions at every scale from the molecular to the ecosystem level, is preserved in amber in a way that no other archive of the past approaches. And now, something about how this amber came to be in human hands. Because the story of Baltic amber and human civilization is itself one of the more remarkable threads in our species' relationship with deep time. The amber trade routes of the ancient world followed what archaeologists call the Amber Road, a series of overland and riverine trade networks running from the Baltic coast southward, through what is now Poland and Central Europe to the Adriatic Sea, and from there by ship to the Mediterranean world. Amber moved along these routes for at least 3,000 years before the Common Era. It has been found in the tomb of Tutankhamun. It has been found in Mycenaean Greek graves. It has been found in Bronze Age settlements across Central Europe, always far from any natural source, always clearly carried over great distances by trade. Why? Because amber was considered magical, because it was warm. Unlike stone, unlike metal, it retains and radiates heat in a way that feels almost biological. Because it was transparent and yet solid. Because it burned with a pleasant smell. Burning amber was used as incense in ancient temples. And because of what was inside it. A piece of amber with an inclusion, with a fly or a beetle or a flower inside, was worth considerably more than plain amber. The ancient world recognized that this was something extraordinary, a creature from the past, perfectly preserved, locked inside a gemstone. They did not have the concept of deep time. They could not have known how old the insects were, but they knew that these creatures had died long ago, trapped in tree resin, and that the resin had somehow turned them into something permanent and precious. That intuition, that amber preserves the past in a way nothing else does, turns out to be one of the most accurate scientific intuitions in human history. It just took us 2,000 years and a revolution in analytical chemistry to understand exactly how right it was. Here is something that Baltic amber has told us that we could not have learned any other way. Inside certain Baltic amber specimens, researchers have found plant material, flowers, leaves, fragments of bark, pollen from the Eocene forest. This plant material represents species that are entirely extinct with no living descendants. But it also represents species that are ancestral to modern plants, ancestors of flowers we have in our gardens today, preserved in amber at the moment when they were alive. In one celebrated specimen, researchers found a flower in the act of being pollinated, a bee or bee-like insect inside the flower, carrying pollen from a related flower, frozen at the exact moment of the pollination event. A moment of ecological interaction from 46 million years ago, captured permanently. Two organisms caught together in a single second, their relationship, the relationship between a flower and its pollinator, made visible and readable across 46 million years. This is what amber does that nothing else can. It captures not just organisms, but moments, events, relationships, the living world in action, stopped at a single instant. But as remarkable as Baltic amber is, it is not the most extraordinary amber on Earth. That distinction belongs to something found much more recently, in a place that has made its study as morally complex as it is scientifically extraordinary. In the Hukaung Valley of Kachin State, in the far north of Myanmar, formerly Burma, there are amber mines. The amber from these mines is approximately 100 million years old. It dates to the Cretaceous period, the mid-Cretaceous specifically, a time when the Indian subcontinent was still an island moving northward through the ancient Tethys Ocean, when flowering plants were just beginning their transformation of the world's ecosystems, and when, this is the part that matters most, non-avian dinosaurs were the dominant large land animals on Earth. The trees that produced this resin were giant tropical conifers related to modern araucarias, the monkey puzzle trees still alive today in South America and the South Pacific. They produced resin in extraordinary quantities, and that resin accumulated in the warm, wet forest environment, trapping whatever came into contact with it, being buried in river sediments and waiting 100 million years through the extinction of the non-avian dinosaurs, through the rise and diversification of the mammals, through the entire evolution of primates, through the emergence of our own species, through all of recorded human history. The amber waited, buried in the rock of northern Myanmar, while everything that happened in this video's previous chapter happened, and more. And then, in the last 20 years or so, it began to come out of the ground. What has emerged from the Burmese amber, the scientific name is Burmite, has been, without exaggeration, the most significant series of paleontological discoveries since the invention of the CT scanner. The inclusions in Burmite are different from Baltic amber in a crucial way. Baltic amber captures the Eocene, a world already recognizably modern in many of its biological features. Burmite captures the Cretaceous, a world in the middle of the age of dinosaurs, before many of the major animal and plant groups we know today had evolved to their modern forms. In Burmite, researchers have found insects from lineages that are entirely extinct, creatures with no living relatives, body plans that evolution tried and then abandoned. They have found spiders with tails, actual tails, a feature no modern spider possesses, found in an amber specimen that immediately forced a reassessment of spider evolution. They have found primitive ants in the earliest stages of the evolution of social behavior. They have found ancient relatives of modern beetles, flies, and wasps, allowing scientists to trace the evolutionary history of these groups across 100 million years with a precision that was previously impossible. But the finds that captured the world's attention were not insects. They were something closer to the creatures most people think of when they think of the Cretaceous. In December of 2016, A paper was published in the journal Current Biology that described something no one had ever seen before. A researcher named Li Xing, working with a piece of Burmite purchased at a market in Tengchong, a town in Yunnan province near the Myanmar border, had found something inside. The piece of amber was being sold as a decorative object, a curio with what the seller thought was a plant inclusion inside. What it actually contained was a section of tail, the tail of a feathered dinosaur, not a bird, a non-avian dinosaur. The tail belonged to a Coelurosaurian, a member of the group that includes tyrannosaurs, velociraptors, and the ancestors of birds, and it was approximately 100 million years old. The tail was small. The entire specimen is less than 1.5 inches long, suggesting the animal itself was perhaps the size of a sparrow. But the preservation was extraordinary. The tailbones were visible. The individual vertebrae were articulated, still in their correct anatomical positions relative to each other. And around the bones, preserved in 3 dimensions, was the soft tissue, flesh, skin, and covering it all, feathers. Not the impressions of feathers, not the chemical ghosts of feathers. Actual feathers in 3 dimensions, with their microscopic structure intact. The individual barbs, the barbules, the hooklets that connect barbules to each other. Feathers from an animal that lived 100 million years ago, preserved as completely as a feather you might pick up from the ground today. The color was even partially readable. Advanced imaging techniques allowed researchers to identify the melanin granules, the pigment-producing structures, preserved within the feather tissue. The feathers were pale or translucent on the underside and darker on the upper surface, in a pattern consistent with countershading, a camouflage adaptation seen in many modern birds and other animals. A 100 million year old color pattern in a creature that saw living dinosaurs. Read from a piece of amber purchased at a street market. If that were the only discovery from Burmite, it would be extraordinary. It is not the only discovery. In 2017, a different team reported the discovery of a baby bird in Burmite. Not a feathered dinosaur, an actual bird, a member of the ancient group called the Enantiornithines, which were the dominant birds of the Cretaceous, and went extinct at the end of it along with the non-avian dinosaurs. This specimen was also small, a nestling found in a piece of amber about 3.5 inches long. But the preservation was almost incomprehensible. Skin, feathers, claws, tissue from the wing, the bones visible through the transparent amber in their correct anatomical positions. A baby bird from 100 million years ago, preserved in 3 dimensions, preserved in color, preserved in detail finer than any fossil bone could provide. The Enantiornithines were extraordinary animals. They were birds. They had feathers. They had wings. They could fly. But they also retained features that modern birds have lost. They had teeth, for instance, actual teeth in their beaks, something no modern bird possesses. They had clawed wings, functional claws at the leading edge of the wing, useful for climbing, that the chick in the amber specimen would have used. They were, in a sense, a bridge between the non-avian dinosaurs and the birds we know today. And we know what they looked like, really looked like, with their feathers and their skin and their proportions. because one of them died in a forest in what is now Myanmar, fell into a pool of resin, and waited 100 million years to be found. But even this is not the strangest thing found in Burmite. For that, we need to talk about blood. But before we get to the blood, I want to pause at one more category of discovery from Burmite that deserves its own moment, because it represents something that changes how we understand the deep history of the groups of animals that make up the majority of multicellular life on Earth. Arachnids in Burmite are extraordinary. Modern spiders number around 45,000 described species, making them one of the most species-rich animal groups on the planet. Their fossil record in stone is sparse. Spiders have soft bodies, and soft bodies fossilize poorly except in very specific conditions. Amber has given us, in the Burmite deposits alone, hundreds of spider specimens of extraordinary quality, and among them are forms so unexpected that they required the creation of entirely new families within the spider phylogenetic tree. The tailed spiders I mentioned earlier, Chimera arachne yingi, described in 2018, belong to a group called the Uraranida, known previously only from Devonian and Carboniferous fossils. The discovery in Cretaceous amber of a representative of this lineage, an animal with the spinnerets and silk-producing capability of a true spider, combined with a long flagellum or tail that modern spiders completely lack, extended the known range of this group by more than 100 million years, and raised profound questions about the early evolution of spiders. Why did some lineages retain the tail while others lost it? What function did it serve? Was the tailed form an evolutionary dead end, or did it persist in some form even later? These questions cannot be answered from the stone fossil record, which provides only fragmentary and deformed impressions of ancient spiders. They can be approached, not completely answered, but meaningfully approached, only because amber preserved these animals in 3 dimensions. The parasitoid wasps in Burmite represent another revelation. Modern parasitoid wasps, insects that lay their eggs in or on the bodies of other insects, where the wasp larvae consume their host, are an extraordinarily species-rich group. Estimated to include hundreds of thousands of species and constituting a major regulatory force in almost every terrestrial ecosystem. Understanding when this parasitoid lifestyle evolved and in what ecological contexts it first appeared is a fundamental question in evolutionary biology. Amber has given us representatives of multiple parasitoid wasp lineages from the Cretaceous, preserved in enough detail to identify their host relationships. We have parasitoid wasps preserved in association with insect egg masses. We have parasitoid wasps with their ovipositors, the egg-laying structures that penetrate host tissue, preserved in the act of insertion into host material. We have, in effect, the crime scene photographs of parasitism events that occurred 100 million years ago. One of the most significant parasitoid discoveries from Burmite was the description of a wasp, Xenocerophron, that appears to represent the earliest known member of the family Cerophonidae, a group of tiny wasps that today parasitize a wide range of hosts. The Burmite specimen was smaller than a sesame seed, Its body, its wings, its legs, its antennae, all preserved in a piece of amber you could lose between the pages of a book. It has been waiting 100 million years to be described. When it was, it pushed back the known evolutionary origin of its lineage by tens of millions of years. This is what amber does to evolutionary timelines. It extends them consistently across every group of organisms that amber has preserved in useful quantities. The amber record pushes the known origin of lineages further back in time than the stone fossil record suggested. This is not coincidence. It reflects the fundamental difference between what amber preserves, the full range of organisms present in a forest at a moment in time, and what stone preserves, a biased and incomplete sample weighted toward large organisms with hard body parts. The amber record does not replace the stone fossil record. It complements it by preserving what stone cannot. And what it preserves, repeatedly, is the message that life was more diverse, more complex, and more modern-looking at earlier dates than the stone record alone implies. Now, the blood. In 2017, another paper described a tick, a tiny, blood-sucking arachnid related to modern ticks, found in Burmite. Not remarkable in itself. Ticks have been found in amber before. What made this specimen extraordinary was what was inside the tick. The tick appeared to have fed recently before becoming trapped in the resin. Its body was engorged, swollen with a blood meal, and preliminary analysis suggested that the blood inside the tick contained red blood cells. Red blood cells from an organism that the tick had fed on approximately 100 million years ago. What had the tick fed on? The context provided a strong suggestion. The tick was found in the same piece of amber as feathered dinosaur material, fragments consistent with dinosaur feathers. The ticks that fed on the blood of Cretaceous animals were trapped in the same resin that trapped the feathers of those animals. The logical inference, not a proven fact, but a scientifically reasonable inference, is that this tick had fed on a feathered dinosaur, a tick with dinosaur blood, in a piece of amber you can hold in your hand. At this point, anyone who has seen Jurassic Park knows where this appears to be going. The conceit of that film, released in 1993 and based on Michael Crichton's novel of the same name, is that dinosaur DNA could be extracted from blood preserved inside amber-trapped mosquitoes and used to reconstruct living dinosaurs. It is one of the most compelling science fiction premises ever devised, and it is, in its specific mechanism, not possible. Let me explain why. Not to dismiss the science fiction, which is brilliant and which Crichton researched carefully, but because the real story of DNA in amber is itself extraordinary, and the reality is stranger than the fiction in ways that are worth understanding. DNA is an extraordinarily fragile molecule. Under ideal conditions, cold, dry, dark, without oxygen, without water, DNA can survive for perhaps 1 million years, perhaps a little longer under exceptional circumstances. We have recovered readable DNA sequences from specimens preserved in permafrost, frozen woolly mammoths, frozen cave bears, up to several 100,000 years old. The oldest DNA ever successfully sequenced came from a horse specimen preserved in permafrost, approximately 700,000 years old. That is our current upper limit, and it required extraordinary conditions: continuous deep freezing, no liquid water, minimal oxygen. Amber is not permafrost. Amber is a beautiful, complex, chemically extraordinary substance, and it preserves morphology, shape, structure, color, even some biochemistry, with astonishing fidelity. But it does not preserve DNA. The amber matrix does prevent immediate bacterial decomposition, which is why soft tissue can survive. But water molecules, the primary agent of DNA degradation, are small enough to penetrate the amber matrix. And even in the absence of bacteria, DNA undergoes spontaneous chemical degradation over time. The chemical bonds break. The base sequences become unreadable. At the timescale of Baltic amber, 46 million years, essentially no intact DNA survives. At the timescale of Burmite, 100 million years, the very concept becomes chemically impossible. The DNA from the amber mosquito in Jurassic Park is fiction. This is not a controversial scientific claim. It is the consensus of every molecular biologist and ancient DNA specialist who has examined the question. The specific mechanism of the film does not work. But here is what does survive in amber, and this is where the science becomes genuinely more interesting than the fiction. Proteins survive. Not always, not completely, but proteins, the complex molecules that do the actual work inside living cells, built from sequences of amino acids, are considerably more stable than DNA over geological time. Some proteins have been detected in specimens tens of millions of years old. Chitin, the structural material that makes up insect exoskeletons, survives extremely well in amber, in some cases with its original chemical structure largely intact after 100 million years. Melanin, the pigment responsible for color in feathers and skin, survives in a form that can still be analyzed to reveal original coloration. And in some cases, fragments of original biological chemistry, compounds from the metabolic processes of the trapped organism, can be detected by sophisticated mass spectrometry. None of this allows us to reconstruct an organism. None of it gives us the genetic blueprint to make a dinosaur. But it allows us to answer questions that no other preservation method can approach. What color were Cretaceous feathers? We can now begin to answer that. What did the immune system chemistry of Cretaceous insects look like? We can begin to approach that. What biochemical defenses did Cretaceous plants use against herbivores? Some of that chemistry is preserved in plant inclusions in amber. And then there is something even stranger, something that, if confirmed by further research, is more scientifically disruptive than almost anything else amber has produced: bacteria. In 1995, a researcher named Raul Cano at California Polytechnic State University published a paper claiming to have successfully revived bacteria from the gut of a bee preserved in Dominican amber approximately 25 to 40 million years old. The bacteria, he claimed, had been in a dormant spore state inside the amber, and when the amber was carefully opened under sterile conditions, and the material from the bee's gut was introduced to a growth medium. The bacteria began to metabolize. They woke up. The paper was controversial from the moment of publication. The primary concern was contamination. The possibility that modern bacteria had entered the amber during the extraction process, and that what Carnot was observing, not ancient bacteria revived, but modern bacteria introduced. This is an extraordinarily difficult objection to answer definitively, because modern bacteria are everywhere. In the air, on surfaces, on skin. And even a single contaminating bacterium, given the right growth conditions, can produce a colony large enough to study. Carnot's work was never definitively replicated to the satisfaction of the scientific community, and the claim of revived ancient bacteria remains disputed, but it was also never definitively refuted. The debate continues, and it points toward a question that amber raises more insistently than any other preservation medium. What is the absolute limit of biological survival? We know that certain bacterial spores can survive extraordinary conditions. Bacillus species have been revived from apparently dormant spore states after decades, after centuries, and in some disputed claims after thousands of years, in environments ranging from ancient salt deposits to the gut contents of medieval-era humans. The upper limit of dormant bacterial survival is genuinely unknown, not because we lack the technology to test it, but because we cannot definitively rule out contamination in cases where the proposed survival period approaches anything geologically interesting. What amber offers, in theory, is the ideal environment for extended dormancy. Stable, chemically inert, physically sealed from the external environment for millions of years. If bacterial spores can survive in amber, and this remains an if, then amber inclusions represent the possibility of reaching into biological time at scales that dwarf anything permafrost preservation can offer. This is not Jurassic Park. This is not cloning a dinosaur. But the possibility that inside a piece of amber purchased at a market in Myanmar, there might be a microorganism that last metabolized when dinosaurs walked the earth, that is, in its own way, more extraordinary than any science fiction premise. Because it might be true. Let me step back from the individual discoveries for a moment and consider what amber is doing to paleontology as a field. Because the transformation is structural, not just additive. Before the intensive study of amber inclusions, our understanding of Mesozoic life was built primarily from what bones and teeth and shells can tell us. This is significant. Bones preserve the basic architecture of animals with hard skeletons, their size, their proportions, the mechanical leverage of their limbs, the structure of their teeth, which tells us about diet. It is an extraordinary amount of information, and the work of paleontologists over the past 2 centuries in reconstructing Mesozoic ecosystems from this material has been one of the great intellectual achievements of science. But bones tell us almost nothing about color, almost nothing about the texture of skin or feathers, almost nothing about behavior, about how animals interacted with each other and with their environment. Almost nothing about the soft-bodied organisms, the insects, the arachnids, the worms, the larvae, that dominated Mesozoic ecosystems numerically, even though large vertebrates dominated it visually, and almost nothing about the plants, because plant material fossilizes poorly in stone. Amber tells us almost all of these things that bones cannot. In the last 20 years, the analysis of Burmite alone has produced the formal description of hundreds of new species, species entirely unknown to science before the amber revealed them. These include new species of insects in dozens of families, new species of arachnids, new species of plants represented by flowers and leaves and pollen. It has revealed body plans that paleontologists had no reason to suspect. The tailed spiders, for instance, belong to a lineage that left no other fossil record and would have been unknown to science if amber had not preserved them. It has shown us that the Cretaceous world was simultaneously more alien, with many lineages that are now entirely extinct, and more familiar than we thought, with many modern-looking forms already present in ecosystems 100 million years old. Every year, the discoveries continue. The rate of publication from Burmite inclusions has been increasing rather than decreasing as more material becomes available and more researchers gain access to it. By conservative estimates, the number of species represented in the known Burmite collection runs into the thousands, and only a fraction of these have been formally described. Somewhere in the amber stockpiles of universities and private collections around the world, there are organisms that will force revisions of evolutionary timelines, that will reveal new behaviors and ecological relationships, that will show us things about the Cretaceous world that no amount of bone digging could ever approach. This should be a story of uncomplicated scientific triumph. It is not. Because the Burmite amber that has transformed paleontology comes from a region of Myanmar that has been under the control of armed groups in conflict with the Myanmar central government for most of the past century. And the mining and sale of amber in the Hukong Valley has, in the view of many scientists and human rights organizations, directly funded armed conflict that has caused enormous suffering to the civilian population of Kachin State. This is not a simple problem. It is not a problem with a simple solution. Researchers who work with Burmite are increasingly required to confront a genuine ethical question. Is the scientific value of these specimens sufficient to justify purchasing material from a supply chain that may fund armed groups engaged in human rights violations? Different scientists have reached different conclusions. Some have stopped working with newly purchased Burmite entirely, restricting their research to specimens acquired before the conflict intensified. Others argue that the scientific value is so extraordinary that the information in these specimens exists nowhere else on Earth and will be lost if the amber is not studied, that continued engagement with the material is justified. Others occupy positions between these poles. I am not going to resolve this debate for you. It is a genuine ethical question, and it deserves to be presented as one rather than flattened into a comfortable resolution. What I will say is that the existence of this debate reflects the reality that science does not happen in a vacuum, that the extraction of knowledge from the natural world has costs, and that those costs are not always borne equally, and that the people who benefit from the knowledge are often not the people who bear the costs. The amber tells us extraordinary things about a world 100 million years old. The amber is also part of a supply chain embedded in a contemporary human tragedy. Both of these things are true simultaneously, and neither one cancels the other out. There is one more thing that amber preserves that we have not discussed, and it is perhaps the most overlooked and in some ways the most philosophically significant of all the material in these deposits: air. When a piece of resin flows over an organism or over a surface, it frequently traps bubbles, small ones, sometimes microscopic, sometimes a millimeter or two in diameter. Bubbles of the air that was present at the moment the resin flowed. Air from the Eocene, 46 million years ago. Air from the Cretaceous, 100 million years ago. Researchers have been analyzing the composition of these trapped bubbles since the 1980s, using techniques that have become progressively more precise. The results have been fascinating and occasionally contentious. Early analyses suggested that Cretaceous atmospheric oxygen levels were significantly higher than today, perhaps 30% or more, compared to today's 21%. If accurate, this would have significant implications for understanding Cretaceous biology. Higher oxygen levels would have supported larger animals, more active metabolisms, faster growth rates. More recent and more careful analyses have been more conservative in their conclusions. The difficulty is that amber itself is not completely impermeable over geological time. Some gas exchange with the surrounding environment may occur, potentially altering the composition of the trapped bubbles. Disentangling original atmospheric composition from post-trapping alteration requires extremely careful methodology. But even the most conservative analyses have provided useful data. The nitrogen to oxygen ratio in ancient amber bubbles, the relative abundances of different trace gases, the presence or absence of certain atmospheric components. All of this contributes to a picture of how Earth's atmosphere has changed over geological time, calibrated against other lines of evidence from geochemistry and paleobotany. You can think of each amber bubble as a tiny sealed flask, corked 100 million years ago. When a researcher carefully punctures an amber specimen under controlled conditions and captures the gas that escapes for mass spectrometric analysis, they are opening that flask. They are breathing, in a very attenuated, analytical sense, air from the Cretaceous. Air that was last in contact with the open atmosphere when Spinosaurus was fishing in North African rivers and Triceratops had not yet evolved. The air is different from what we breathe. Not dramatically, the broad composition of Earth's atmosphere has been relatively stable for hundreds of millions of years, but measurably. The CO2 levels were higher, consistent with the warm Cretaceous climate. Some trace compounds reflect a different biosphere, a world aoi with different dominant plant groups producing different volatile organic compounds. A world before the great diversification of flowering plants had fully transformed the chemistry of the air. This is the smallest of amber's preservations, and perhaps the most intimate. Not a creature, not a landscape, not a moment of behavior frozen in time. Just air. just the atmosphere of a vanished world, trapped in a transparent stone, waiting for us to open it. You can think of each amber bubble as a tiny sealed flask, corked 100 million years ago. When a researcher carefully punctures an amber specimen under controlled conditions and captures the gas that escapes for mass spectrometric analysis, they are opening that flask. They are breathing, in a very attenuated, analytical sense, air from the Cretaceous. Air that was last in contact with the open atmosphere when Spinosaurus was fishing in North African rivers and Triceratops had not yet evolved. The air is different from what we breathe. Not dramatically, the broad composition of Earth's atmosphere has been relatively stable for hundreds of millions of years, but measurably. The CO2 levels were higher, consistent with the warm Cretaceous climate. Some trace compounds reflect a different biosphere. A world with different dominant plant groups producing different volatile organic compounds. A world before the great diversification of flowering plants had fully transformed the chemistry of the air. But the atmospheric analysis of amber bubbles goes further than just oxygen and carbon dioxide. Researchers have detected traces of ancient volcanic emissions, sulfur compounds from Cretaceous volcanic activity that left their chemical signature in the air and that the amber captured. They have found organic compounds that reflect the specific plant communities of different amber-producing forests. The chemical fingerprint of a Cretaceous conifer forest in Myanmar, distinct from the chemical fingerprint of an Eocene subtropical forest in what is now northern Europe. Amber is not just a biological archive. It is a chemical archive of the atmosphere of the past, the smallest of amber's preservations, and perhaps the most intimate. Not a creature, not a landscape, not a moment of behavior frozen in time. Just air. Just the atmosphere of a vanished world, trapped in a transparent stone, waiting for us to open it. And this brings me to something that has happened in the last 5 to 8 years specifically. A wave of discoveries so concentrated and so consequential that it has changed the pace of the field entirely. The development of synchrotron X-ray computed tomography, a technique that uses extremely intense X-ray beams generated by a particle accelerator to produce three-dimensional images of the interior of objects at microscopic resolution, has transformed what researchers can see inside amber specimens. Before this technique, studying the internal structures of an amber inclusion required physically cutting the amber, which destroyed the specimen's context and risked damaging the inclusion itself. With synchrotron CT, researchers can produce complete three-dimensional maps of everything inside an amber specimen. The organism, its internal anatomy, any associated material, without touching it, without opening it, without disturbing anything. What this has revealed, in specimen after specimen, has repeatedly forced revisions of what scientists thought they knew. In 2019, synchrotron imaging of a Burmite specimen revealed the earliest known example of brood care in insects, a mother beetle trapped in amber 100 million years ago, surrounded by her eggs. The eggs were arranged in a specific pattern consistent with deliberate placement. The mother was oriented toward them. The inference, carefully stated by the researchers with appropriate scientific caution, is that this beetle was engaged in active maternal behavior at the moment of entrapment, a behavior 100 million years old, readable from an amber specimen using a particle accelerator. In 2020, a team led by researchers at the Nanjing Institute of Geology and Paleontology described the oldest known representative of the insect order Neuroptera, the lacewings, in a Burmite specimen. The specimen was so well preserved that the researchers could identify individual eye facets in the compound eye, map the venation of all four wings with complete accuracy, and determine the precise body posture of the animal at the moment of death. This level of anatomical detail from a 100 million year old specimen is simply impossible from any other preservation medium. In 2021, multiple specimens of a completely new family of parasitoid wasps were described from Burmite, wasps that parasitized spider egg sacs, a behavior known from a handful of modern wasp species. Finding the ancient representatives of this lineage preserved in the act of their parasitism, one specimen was found inside a spider egg sac, revealed that this behavior has existed for at least 100 million years. An ecological relationship between wasps and spiders, preserved not as inference from anatomy, but as direct physical evidence of the interaction. And in 2023, perhaps the most visually extraordinary discovery yet, a piece of Burmite containing what appears to be an entire ecosystem captured in a single specimen. A piece of amber approximately 2 inches long containing a flower, a bee-like insect in contact with the flower, fungal material on the flower's stem, and a tiny predatory spider positioned nearby. 4 organisms, 4 ecological roles: pollinator, plant, decomposer, predator, caught simultaneously in a single moment of ecological interaction 100 million years ago. A snapshot, not just of individual organisms, but of a functioning ecological community. Every year, discoveries like these are announced. The pace is not slowing. The amber is not running out. The analytical techniques are improving, and the organisms inside the amber have been waiting patiently for 100 million years. They can wait a little longer while we develop the tools to read them properly. Now let me bring this story to its conclusion, because there is a larger question underneath everything we have discussed, and it is the question that amber poses more insistently than anything else in the fossil record. What does it mean to hold the past? We have a complicated relationship with time. We experience it as moving, as something that flows past us in one direction, carrying events away from us and depositing them in the inaccessible past. Memory gives us an imperfect purchase on what has passed. Photographs and recordings give us a slightly better purchase. Archaeology and paleontology give us an indirect and fragmentary purchase on the deep past. Amber gives us something that none of these other methods approach. It gives us the thing itself, not a record of the thing, not an impression of the thing, not a chemical trace of the thing, the thing. The organism in 3 dimensions, in the form it had at the moment it died, sometimes with its original color, sometimes with traces of its original chemistry, sometimes with its ecological context, the plant it was sitting on, the organisms it was interacting with, the air it was breathing, all captured simultaneously in that 1 second when it touched wet resin. There is something about this that is philosophically disorienting in a productive way. When you look at a bone fossil, you are aware of the enormous gulf between you and the organism that produced it. The bone is stone now. The animal is utterly gone. The time between is a void you cannot cross. But when you look through amber at an insect that died 100 million years ago, at its compound eyes, at the texture of its wing surface, at the posture it happened to be in at the moment of death, the time collapses. Not figuratively. There is no impression of distance, no sense of the void. The creature is simply there, the way any living creature is there when you look at it. The only thing separating you from it is a layer of ancient resin, and the 100 million years that layer represents are invisible, unfelt, almost impossible to internalize. This is not just an aesthetic observation. It has practical implications for how we understand the history of life. Amber specimens from the Cretaceous are being studied using techniques that did not exist 10 years ago. Synchrotron X-ray imaging, which can produce three-dimensional maps of internal structure at microscopic resolution without damaging the specimen. Fourier transform infrared spectroscopy, which can identify original biological chemistry preserved in the amber matrix. Confocal microscopy, which allows optical sectioning of amber specimens to reveal three-dimensional structures without physical sectioning. These techniques are allowing researchers to see things in amber that no previous generation of scientists could see. And there is every reason to expect the techniques developed in the next decade will reveal things we cannot currently imagine. Every piece of amber is a message from the past. Not a translated message, not an encoded message, not a message that requires significant interpretation to decode. A direct message in the original language, written by the organism itself in the form of its body, sent 100 million years ago and received today. We are the first generation of humans with the tools to fully read those messages. And what we are reading is extraordinary. A feathered dinosaur with its colors readable. A baby bird from a lineage entirely extinct. A tick with what may be the blood of the animal it fed on. A spider with a tail. An insect pollinating a flower. Air from the Cretaceous. Possibly, microorganisms that last metabolized when the continents stood in different positions. The evidence points to one conclusion. Amber is the most important physical archive of the history of life on Earth. Not the most comprehensive. Bones and teeth and shells preserved in rock tell us more, in total volume of information, about the history of vertebrate life. But the most important in the specific sense that amber preserves what nothing else preserves, answers questions that nothing else can answer, and does so with a fidelity and a directness that is simply without parallel anywhere in the fossil record. I want to be more specific about what I mean by that. because I think the implications are worth sitting with. Every major revision in our understanding of what Cretaceous animals looked like, what color their feathers were, how their soft tissues were arranged, what behaviors they engaged in, has come, in the last decade, substantially from amber. The revolution in our understanding of feathered dinosaurs was driven primarily by the spectacular fossils from Liaoning province in China. Yes, but the specific details of feather microstructure, of color, of the fine anatomy that tells us about flight capability and insulation and signaling, those details come from amber. The bones told us the architecture. The amber told us the appearance. Every major extension of the known evolutionary history of insects, the group that comprises the majority of all animal species on Earth by any measure, has come from amber. The stone record of insects is sparse and fragmentary. The amber record is dense and complete. When researchers want to understand when a lineage first evolved, when a behavior first appeared, when an ecological relationship was first established, they look at amber. And every time a new analytical technique becomes available, synchrotron imaging, high-resolution mass spectrometry, confocal microscopy, chemical imaging, the first place researchers apply it is to amber specimens, because amber is where the answers are. It has been waiting for the questions for 100 million years. There is something humbling about this, clarifying rather than diminishing. We have spent the last 2 centuries building an increasingly detailed picture of prehistoric life from bones and impressions in stone. And that picture is extraordinary, a genuine triumph of scientific inference. And then amber comes along and shows us, directly and without inference, that the picture was right in its architecture and incomplete in its detail. The bones correctly described the structure. The amber fills in everything else. Together, they give us something approaching a real portrait of vanished worlds. Not a silhouette, not a sketch, a portrait, with color, with texture, with behavior visible in the posture of the moment of death. The amber does not judge what it preserves. It does not distinguish between the significant and the incidental. It captures whatever it captures, holds it for however long it holds it, and offers it to whatever intelligence eventually finds it and learns to read it. This indiscriminate fidelity is both its limitation, we cannot choose what amber preserved, and its greatest strength. The amber record is biased by the accidents of which organisms happened to touch resin in which forests, but within that bias it is perfectly honest. What you see through amber is what was there. Imagine that insect again, in your hand, warm, transparent, the legs folded, the eyes intact. 100 million years old and looking, in some sense that is not quite metaphorical, at the same world it was looking at when it died, just from a very different angle. That is the strange world of amber, a machine made not of metal and electricity but of tree sap and time. A machine that does not move us through time but brings time to us, intact, readable, extraordinary.